Hi everyone, this is Dr. Santosh. Today we'll talk today about structure of DNA and the various form of DNA. So let's see first V DNA and we'll delve into the very detail of the DNA. So you can see this V DNA, this structure you can see here. This is a polymer of nucleotide. So DNA is polymer of nucleotide, deoxynucleotide. And you can see here it is between this is one nucleotide, this is second nucleotide, this is third nucleotide, and this is fourth nucleotide. Between nucleotide you can find the phosphate. The phosphate will join this bond. So here two nucleotides are joined by deoxynucleotide. Deoxy means the oxygen is not there. Second position of this base you don't find here oxygen. So this is deoxyribose. Deoxyribose nucleotide is bound with the phosphodiester bond. What does that mean, phosphodiester bond? You can say this is the two oxygen is bond here and this is double bond. So this is a diester, one ester here and other ester here. So this is phosphodiester bond. And if you notice, you can see the phosphate is present here. It is attached to the 5' region and we call it as head. This is direction. When you see direction of DNA, we call this part as head. And now when you see the last part as tail, you find if it is an end, you will find OH group will be there. Okay, so head will have phosphate group and the tail will have OH. So the direction of this polymer is towards 5' phosphate to 3' this OH group. So the direction of this DNA polymer is from 5' to 3'. And this DNA molecule you can find it is a highly polar molecule. And in terms of directionality also, and you can see the molecule contain lots of phosphate backbone. So and the sugar backbone itself is also somewhat hydrophilic. So phosphate sugar backbone makes this whole backbone, especially phosphate, makes it polar molecule. So this is the single strand. If you see the single strand of this DNA, it is polymer of nucleotide, polar polymer of nucleotide, which have a directionality from 5' to 3'. And this DNA is a double-stranded molecule. So double-stranded molecule means it has one strand here. And this strand is complementary. And complementarity is gained because of this base pairing in the middle at the core of DNA. So this base pairing, you can see the G and C base pairing is there are 3 hydrogen bond. Hydrogen bond is a non-covalent bond. The attraction, it is just a attraction between the hydrogen, partially positively charged hydrogen, and with oxygen, that is negatively charged, or other electronegative, for example nitrogen. Here the hydrogen will form the bond with the oxygen. So this is hydrogen bond. This is non-covalent bond. That means it is like an attraction between this partially charged molecule. Now you can see in thymine and adenine, here guanine and cytosine, there are 3 hydrogen bonds. In thymine and adenine there are 2 hydrogen bonds, and between cytosine and this guanine there are 3 hydrogen bonds. Again thymine and adenine there are 2 double bonds. So this is complementarity. 2 complementary polar DNA molecule are placed in anti-parallel direction. You can see 5' to 3', here also 5' to 3'. You can find this here. So this is the direction here, 5' to 3', and then 3' to 5'. So if we have to summarize, we say DNA is a polymer of nucleotide. Double strands are polar and placed in anti-parallel direction, and they will form right-handed helix. So when we see right-handed helix, we put our right hand like this and we see the helix like this. So DNA is the balanced DNA. The DNA that was discovered by Watson Crick is the right-handed helical model. Okay, so this is the structure of DNA, and you can see here 10 bases are present per pitch from here to here. So 10 bases are present per pitch, and in that base in that DNA there is major helix and minor helix. My understanding of major helix and minor helix is a little tricky. I have already done video, but major helix, you can see there is alternating major and minor helix. You will understand this structure when you straighten this DNA, whole molecule of DNA from helical to straight ladder or stair like fashion. If you unfold this DNA, the one face of the stair will be major group, other face of stair will be minor group. So this is the whole DNA. This is the measurement of DNA. DNA is a 2 nanometer width and it has got 3.5 nanometer length per pitch. Per base the length of this DNA is 0.34 nanometer. This is the length of DNA per base. Between two bases, the length is 0.34 nanometer. So this is the structure of balanced DNA, and we will understand this structure of balanced DNA when we understand other structure well. And when we compare this balanced DNA B DNA with other structure, we understand this B DNA very well. One thing, and we will also understand that this B DNA is not exact representation of the native DNA that is present in our body inside the nucleus. So it is not representation complete. It is not exact representation. It is average representation of the native DNA. The native DNA is very close to this B DNA. So let's see the other DNA structure. Now, okay, let's see other DNA structure. You can see here B form of DNA, which is observed in high humidity. That is, high humidity means the cell has very high water content. So this B DNA is present in cellular environment. So B DNA is approximate. It is average structure of the DNA. B DNA is the closest form of DNA to our native DNA or physiological DNA. So it is observed in high humidity because when DNA structure is analyzed, it is analyzed in through X-ray crystallographic method, and so we need to take out that DNA outside, and then we need to see that DNA. So when you put the DNA in high humidity environment, then you will get V DNA, and which is very close to our main DNA structure. Must closely correspond to average DNA structure under physiological conditions, but it deviates in two respects from DNA in cell. You can see that DNA, B DNA, we have seen there is 10 bases, but in DNA that is present inside the cell, it is a little more twisted. That means it has got more bases per turn. It has 10.5 bases per turn. And then real DNA is not perfectly regular. So DNA will have some base coming out, popping out, or somewhere it will not be exactly 10.5, somewhere it will be less. So little bit of variation is there. To understand that variation also, we need to go over other structures of DNA. Then we will have a composite image of DNA in our brain. So you can see, and this real DNA is not perfectly regular. That also means when you see the bases of real DNA, we see this bases is very much parallel to the when you take the base and pull it, make it vertical, the whole DNA. If you make the whole DNA vertical, when you see this bases in the model, double helical model of Watson-Crick, you can see the base, they are very parallel to the floor. But in reality, this base is not parallel to this floor. You can see there is twist. This is called propeller twist. This base will propel each other, and this is called propeller twist. You can find in the native DNA. So this is the real DNA. Real DNA deviates from the DNA P DNA, but it is the B DNA is the most closest form of the DNA of our body. Now let's see the alternative form of DNA. Alternative form of DNA are aDNA and zDNA. And aDNA, you can find aDNA in some places in our cell also, when there is protein and DNA interaction, when that protein make that DNA more dehydrated kind of thing, when that protein place that DNA into more dehydrated condition, so it will be, the DNA can take a form of aDNA. And second important thing is this aDNA, when RNA, it folds into double helical structure. RNA also folds into double helical structure upon itself. Okay, the same RNA molecule will fold and will form double helical structure. When RNA is double helical, this RNA, the main form of RNA is A form. Double helical structure of RNA is A form. So that is why A form, it is important to understand about A form also. So you can see most RNA is A form, RNA DNA duplex is in A form, and A form is present when there is dehydrated condition. So how I remember is this: A is for anhydrous and achondroplasic. The strand, the double helical structure, little stunted. The size between this one pitch is little less here. So that is why I call it achondroplasic, means dwarf DNA, and this is anhydrous DNA. So this is thing. These two A will help me remember how aDNA looks like and which condition we get aDNA in. And Z-DNA actually you get a Z-DNA in very high salt concentration. Okay, but when sodium will interact with the phosphate backbone of the DNA, the DNA will conform into Z-DNA. Z-DNA is a left-handed DNA. Okay, Z-DNA is left-handed. The significance of this DNA is in that the Z-DNA, there is mechanism inside cell, especially in some lower organism, eukaryotic organism. The mechanism has been found that there has been seen that twisting of DNA is associated with the regulation of G. So this Z-DNA, though real significance has not been yet determined, but they consider that Z-DNA might be very important for the gene regulation mechanism in certain organism. So this is the main significance of knowing DNA. Let's delve into detail of this Z-DNA and A-DNA, how they deviate from this B-DNA. To understand A-DNA and Z-DNA, just again let me see the relation of this bases with sugar in B-DNA, sugar and phosphate bond. This is red line, you can consider this as sugar, and this you can consider it a phosphate or sugar phosphate backbone, and this line you can see is a part of sugar. So sugar and N-glycosidic bond, you can consider it like that. So when you straighten this whole duplex, double helical structure of DNA, when you make a stair, like looking like a stair, so what you see, you will find the bases are placed in relation to sugar or phosphate like this. This N-glycosidic bond will not divide this whole base equally. Rather, N-glycosidic bond will form this type of angle. This part of this whole, if you take this as circle, this whole 360-degree circle, and if you see this part, this will be minor group. Okay, one phase will be minor group, other phase of this whole DNA molecule, when you straighten this molecule, this will be major group of this DNA. Now once you understand major and minor group, what happens in other form of DNA, this other form of DNA, they have actually distortion of this structure, this ideal structure of major and minor group. They have little bit distortion of this, and there is a problem. This other DNA will have altered, they deviate from this B DNA in relation of this sugar phosphate bond with the base. That is the main difference, the most important difference. Now let's see this ADNA. What happens? Now you can see the ADNA, the base will pop out, and you can see this is the minor group. This minor group will become very wide in ADNA. And so that is why you can see this minor group will be outside and major group will be inside. Okay, let me first talk about Z-DNA also, then we will talk about how this base is placed in double helical structure in helix. Now let's see this Z-DNA. When you see Z-DNA, this minor group, if you compare this Z-DNA with the B-DNA, so you can see minor group is constricted more and major group has popped out more. And you can notice one more thing. When this base is pure, so you can see this will be folded like this. So this phosphate and sugar will be folded in the same direction. Okay, so this type of arrangement is called syn arrangement. Same direction, same way it is folded. When this with purine, the base sugar, the relation of sugar and base with purine, relation of deoxy sugar with purine is like this. It will be folded, but it will not be that folded. It will be straight. This is called opposite. So for sugar and the base for pyrimidine. So for purine it is syn, and that is very important for this Z-DNA conformation. And very important thing to remember for all of this, the major group and minor group. You can see the major group is very wide here, very wide. Okay, the major group is narrow in ADNA, and major group is very shallow and wide here in Z-DNA. Minor group is constricted more than this B-DNA. So actually the base will shift towards major group. Here base has shift in ADNA, shift towards minor group. So this is the conformation, and this type of conformation, this stair conformation, now take the round. It will take the helical round. So let's see how it forms helical. So let me first talk about this aDNA here. Okay, now aDNA, you can see the aDNA base will be pushed outward. aDNA base will be pushed outward. We will see that how base is pushed outward in the animation. So you can see that when bases are pushed outward, and this part, you can see bases are pushed outward at this part, at the center is major groove and the periphery will be minor groove. So when you see the bases will be rotating like this in right-handed helical fashion, and bases will be outward. While you see, when you see in bDNA, but the bases will be at the center. The axis of this whole DNA, you can find this base will rotate along the axis. Okay, and in aDNA you can see the base rotates around the axis, and this axis lies in this major group. This whole middle center is a major group. So axis of this DNA, when you see the DNA from the top, you find the axis. So this axis will lie in major group. Here axis is in goes through the base. You can see this is how base will rotate here. So this is the bDNA. But in zDNA, it is a left-handed fashion, and then the repeat of base will be the 2 base will repeat like this. So you can say this is the base repeat like this. 2 bases will repeat in left-handed helical. So you can say this is, if you see here, I have shown this DNA, zDNA. When you see from the top, the cross section of bDNA is zDNA is constricted. zDNA is the most constricted, but I have shown you, I have to show this base pair rotation. That is why I have shown this very wide. But understand that this base is not that wide. If you compare with this, the zDNA is the narrowest cross section. It has got narrowest cross section. And when you see aDNA, you can see there is empty space in the middle, and the base is pushed away at the towards periphery. Somewhat base is also pushed here in the periphery in zDNA also. It's not the base does not rotate along the rotate the center of the axis does not pass through the base. So base will also be little bit pushed towards the outside, but you can see it will push towards the major groove, and here in aDNA it will push towards the minor groove. So this is, if you remember this very nicely, it will help you understand and memorize also aDNA, zDNA and bDNA. This is the structure of aDNA, zDNA and bDNA actually in relation to base, how base shift, and this is the most important part of everything, this structure. Now let's see this DNA conformation. Now let's see the dimension of various types of DNA. So when you see the dimensions of various types of DNA, there are many numbers. It looks very confusing for you. Now you have to make a strategy to see the DNA, everything. The DNA, if we see the base pairs, we look at the base pairs. We have already looked at the base pair. Actually, we have idea how this base pair are present when we look the DNA from the top to the axis of the DNA. So when we see the DNA from the top or bottom, then we see the base pair rotation. So this all characteristics signifies this base pair relation. That now this is the dimension measurement of the DNA. I told you the A is achondroplagic. I told you that A is achondroplagic. That means it is stunted dwarf DNA, and B is a balanced type of DNA. C is we will compare this. C is the leanest DNA. You can say zigzag, and it will have leanest. This is the leanest DNA, because you know that cross section we have seen is the thin cross section. The cross section is very lean. So when you see this is the leanest DNA, and it's the dwarf DNA and thick DNA. Now when you see the group and major group and minor group, I have already told you what is major group and minor group, and I have shown you where axis pass through. So this is the description of major group and minor group. And bDNA, if you see the bDNA has narrow minor group and wide major group. But when you see at aDNA, I told you aDNA, the base is the 2 bases are pushed towards the minor group. So there you will find the minor group is broad and narrow, but major group is broad and shallow, because the base pair is pushed towards minor group, and then major group will become narrow. So this major group is narrow in a helix and this wider in the minor group is major group is narrow in ADNA, and minor group is shallow and wide. So you can find this. Now, in Z-DNA, when you see major and minor group, I told you the major group, if you can remember the picture, the major group, this whole base is pushed towards minor group. So pushed towards major group. The whole base is pushed towards major group. So you can see the characteristic will be major and minor group of in Z-DNA. Characteristic will be very similar to this. So you can see rather than accentuated characteristic, you can see in V DNA, the major group will be flattened, more flattened, and minor group will be more narrow. Here opposite, ADNA, the major and minor group is opposite. Now when you see the base turns, because this is base turn, if you see almost there measurement wise, there is 11 bases per turn, 10 bases per turn, and if you see rotation also per base, if you divide this 11 by 360 degree, so you get the base rotation per turn. So you get obviously base rotation per turn will be more in BDNA than, and here base rotation is in anti-clockwise. Sorry, it is in left-handed. So ZDNA is left-handed helix. ZDNA is left-handed helix. So you can see negative turn of two base pair. So per turn there will be two base pair will turn simultaneously. So this base turn, and you can find this axis. This signifies how base is positioned towards the horizontal floor. If you put the whole DNA as in vertical, so you will see the base is more inclined here, and almost base you will see the base is almost flat. Base is almost parallel to the surface, and here also it is more parallel to the surface in comparison to this ADNA. ADNA you will see base is very inclined to the floor, and base pair propeller twist is also somewhat similar between this B and ZDNA. It is a propelling characteristic of two bases. These two bases will not be in very perfect parallel configuration. They will propel each other. So that is what you see here. Now here you can see. So this is what you can see in ADNA. I told you this is dwarf DNA. So you can see short and broad DNA, and the same thing you can find in this also. So there you can find the rise is very, because it is short, so distance between two bases is also less. This is somewhat like you can consider like a length of this DNA. The length of DNA is space between base and between this helix. So this is dwarf DNA. So length characteristic of this DNA will be low, but the breadth characteristic, the thickness of this DNA will be high. So you can correlate with it. Actually you don't have to memorize, but if you see the figure you can correlate with the structure of DNA. Both are right-handed, only cDNA is left-handed. What is very important thing about this cDNA is this anti-acid configuration. So you will see all DNA have anti configuration of this sugar with base. So anti and syn is the relation of sugar with the base. So they have an anti configuration, and here you will find this anti for B, C, and syn for G. So that makes this DNA somewhat linear DNA and zigzag DNA and left-handed DNA. So this is the characteristic of these three forms of DNA. Now you understand this B DNA more nicely. You understand this B DNA well when you compare this DNA with others. Now let's see the transformation of B DNA to A DNA. So this is in animation, you can see this B DNA. Now B DNA, you can see the base is somewhat parallel to the floor. When you take the DNA, you put the DNA in vertical direction, and you can focus on this sugar also. If you can focus on sugar is somewhat perpendicular to the floor, or you can see the parallel to the paper. You can see this. When you see this sugar is very perpendicular to the bases, and this is the structure, and so these bases are placed at the center of the molecule. If you see from the top, it is placed at the center. Let's see the whole DNA from its top view. Now you can see DNA from the top, this P DNA from the top. You can see this base is placed at the center. Okay. Now let's see what happens in aDNA. So in aDNA, you will see Now here you can see this is the minor group, okay, and here you can see this is the major group. Now let's see how this base shift towards the minor group, and major group will be narrow. See this, you can see this is now major group become narrow, and this whole base is pushed towards the minor group. Again, let's see this difference. Or you can see this animation now. This is V DNA and this is A DNA. So you can see this. You have seen the shift of the base. Base is present at the center, and then major group is wide and minor group is narrow in V DNA. In A DNA, you will see major group become narrow and minor group becomes wide. So this is the difference between V DNA and A DNA when you see the DNA from the top. See A DNA, you can see the A DNA. These bases are pushed outward and these bases are not parallel, they are in angle with the floor. But while in V DNA, the bases are at the center and they are somewhat parallel to the floor. This is the main difference between A DNA and V DNA. And this happens because when you place DNA into anhydrous condition, when you take moisture from the DNA will take this conformation. So you can see how DNA conform from B to A. You can see this is V DNA. Now V DNA will conform into A DNA in dehydrated condition, anhydrous condition. In anhydrous condition, you can see how this B DNA, you can see how this B DNA conform into A DNA. Now here you can see Z-DNA. Now Z-DNA is left-handed, it has got left-handed helix. You can see the rotation of this Z-DNA, and this is a left-handed helix. You can see if I rotate a little bit, you can find this is a left-handed helix. And left-handed helix, and if you can see this structure very carefully, you can see this is you can find two bases. Two bases will be rotating in pair of two bases. You can see this two base will come above this two base. The two bases will form angle between these two bases. So base repetition happens in chunk of two bases, and then this is a left-handed helix. You can see the helix like this, left-handed helix. And a very peculiar feature you will see here is, if you focus here on this, if you really see closely this two bases, you can see this is pyrimidine attached to the sugar. So this is pyrimidine and this ribose sugar, this deoxyribose sugar, they are in anti-configuration. But when you focus on this pyrimidine, other pyrimidine, you can see the sugar is bent little bit. So this is syn configuration. This is syn configuration here with purine. Look at this syn configuration. This purine is like is very close to the purine. This sugar is close to the purine. That means sugar is folded towards the purine. But when you see the pyrimidine that has formed the base pair with this is not folded. This is straight. The sugar is in anti-configuration. This is very peculiar thing you can find in Z-DNA. That is the reason that Z-DNA, when you see from the top or bottom, it is very constricted. It will look like the diameter is less than the V DNA, and then structure. And one more thing you can notice here also, you can see the bases, they are pushed towards periphery like V DNA, like A DNA. The bases are pushed towards periphery like A DNA here also. But this basis is pushed towards the major group, not minor group. In V, it is pushed towards the minor group. In A-DNA, it is pushed towards the minor group. So thank you. I hope that these all things will help you understand the V DNA structure really well, and it will also help you understand other forms of DNA. Thank you.